Let's look at some of those parts of a cell. In order to start to study a cell, that smallest unit of life, we really need to focus in on the concept of a plasma membrane. Now, a plasma membrane is mostly going to be made up of phospholipids. And as we left off with in our last chapter, phospholipid is a lipid. And it's a really, really interesting lipid because it's got two main parts. It's got this hydrophilic, water-loving head that is really attracted to water. And we've got these two hydrophobic tails. One hydrophobic tail is a saturated fatty acid chain, so it is straight and it is stable. The other fatty acid chain has a double bond between the kinks, making this fatty acid chain unsaturated. It makes that molecule kinked. So within one individual phospholipid, we have a tremendous amount of molecular tension. Part of that molecule is looking for the water. It's attracted to water. And the other part of the molecule is going, ew, ick, I hate water. And within those hydrophobic tails that are repelling water, one tail is straight and a saturated fatty acid chain and very, very stable, while the other fatty acid chain is kinked, has that double bond, and is a little bit more fluid. So we have a lot of molecular tension. This always makes me uncomfortable every time I talk about it because I know that phospholipids coming together to make a plasma membrane can make something like the cell membrane. The cell membrane is a type of plasma membrane that's going to define the inside and the outside of the cell. It's going to be that boundary layer. Cells can control their interaction with the environment through the plasma membrane, and without that cell membrane, that plasma membrane, all of those phospholipids and other molecules interacting, without all of those molecules, we don't have an actual cell. And so when we look at a plasma membrane, like the cell membrane, each individual phospholipid has a lot of molecular tension. And then those phospholipids are going to bond together at the tails, at those hydrophobic tails. And essentially what this forms is a bilayer. So all cell membranes have a bilayer that's just two phospholipids thick, and all of those phospholipids are constantly wiggling and jiggling. And if we were in a classroom together, I would have us all stand together in a group, real close but not touching, because our phospholipids never quite touch exactly. They just get really, really close to one another. I'd have us all stand really, really close to one another, and everybody just kind of wiggles a little bit, and that's exactly what's happening with phospholipids. Now, when I look down at all of these cells that make up the skin of my hands and my arms and my entire body, I have to come back to this understanding that the only thing that's separating me from the rest of the world are two phospholipids. And then within this plasma membrane, within my cell membrane, I know there's going to be all kinds of different proteins. And some of those proteins, like channel proteins, just make a great big hole. And so then I look down at these cells of my skin and I realize that they've got a whole lot of holes. And they're interacting with the air around me and the entire environment around me. And then when I go out into a group of people, I get really, really squeamish because I know that all of our cell membranes have holes, that we're all interacting with each other. We're sharing what looks invisible, but we're sharing all of this space and this air and these molecules between us. So anyways, I'm starting to get a bit off topic, starting to get too big picture. Let's take a look here at our plasma membrane. Right now our plasma membrane just has phospholipids. What we're concerned about, what we're interested in, is understanding how cells are going to control their interaction with the environment. In other words, how they're going to get stuff in, how they're going to get stuff out. And one of the things that cells will have in their plasma membrane is they'll have something like a great big pump protein. And a pump protein is going to be a specialized type of protein that's going to use active transport. It's going to use ATP energy to move things back and forth across our membrane. So I'm just going to draw this big red blob, and this is going to be my pump protein. So our pump protein is going to break a molecule of ATP. When it breaks a molecule of ATP, it's going to release energy, and it can push a molecule across that plasma membrane. So another type of movement that we're going to see back and forth across a plasma membrane will be simple diffusion. And there are two molecules in particular that cells are going to let come and go as they please through simple diffusion, and one of those is oxygen. Oxygen can just slip through the plasma membrane, slip right between those phospholipids. It can come and go as it pleases. The other molecule that's going to be important and that cells will move according to simple diffusion will be carbon dioxide. I'm going to do carbon dioxide in blue. So carbon dioxide can just slip between those phospholipids. Carbon dioxide can come and go however it pleases. So one of the things that we look for in science is we look for these really big themes, or we look for these consistencies. Simple diffusion requires no energy. It's the easiest way to be able to move molecules. So if cells are moving oxygen and carbon dioxide according to simple diffusion from an area of high concentration to low concentration without expending any energy, that starts to tell us how important these two gases are to our cell. Our cell doesn't want to control these. It needs carbon dioxide to get out if we're talking about animal cells. It needs oxygen to get in. We don't want to interfere with this. We don't want to slow it down. We want this to happen as quickly as possible. And so this is one of the reasons why cells will allow carbon dioxide and oxygen to come and go according to simple diffusion. So some molecules and atoms we're going to want strict control on. We're going to use active transport and typically use a pump protein, expend some ATP energy to move those molecules back and forth. Other molecules like oxygen and carbon dioxide can come and go according to simple diffusion. But let's look at the case of water. Now remember, water is two hydrogens bonded to an oxygen. We have that covalent bond, that unequal sharing of electrons. This causes our water molecule to be polar. That's going to be important. And we all know, or hopefully are familiar with the idea that cells need water. Human cells are mostly water. They're like 99.9% water and just 0.1% stuff. Carbohydrates, proteins, lipids, nucleic acids, organelles like the mitochondria and the Golgi apparatus, ribosomes, all of that fun stuff. So if our cell is mostly water, and it goes without saying that water is hugely important to a cell, why aren't cells allowing water to just come and go according to simple diffusion? Well, part of that is because of the structure of our plasma membrane. When we have our two phospholipids bonded together, these tails in the middle make sure that no water can get through. So automatically, with this three-dimensional structure of our plasma membrane, we're going to need something to help water get across that hydrophobic tail barrier. The other thing is that sometimes we need molecules like water to move faster than just simple diffusion. Almost looks like they're violating the laws of physics. So, what our cells have is a special protein that's going to manipulate the polarity of water. And by manipulating the polarity of water, it's going to make the water move in and move out much faster than expected. So this is facilitated diffusion. The example that I have in the lecture slides is of the aquaporin. Aqua meaning water, porin meaning pore. So our aquaporin is a channel protein. And what I've drawn here is I've drawn that aquaporin as if I've sliced it in half. So we have to imagine it as being like a three-dimensional kind of cone, and that three-dimensional kind of cone sits in the cell membrane, sits in the plasma membrane. But I want to cut it in half because I wanted to emphasize this concept of facilitated diffusion and how our cells are able to move water faster than just simple diffusion. So with our aquaporin, the outside edges are more negatively charged. The middle part of our protein is positively charged. So, remember our water molecule has our 2 hydrogens and an oxygen. And so we get this kind of Mickey Mouse shape. This shape is important in addition to the polarity of the water molecule. So what happens is, as our water molecule gets close to our aquaporin, that positive hydrogen gets attracted to this negative part of our protein. Our water molecule then flips as the oxygen gets attracted to the positive middle section of our protein. And then our water molecule flips again as the hydrogen get attracted here to the other negative part. So, if our water is moving in, it can flip, really, really fast, really quick. Make that molecule move faster than simple diffusion because of the polarity of the aquaporin, manipulating the polarity of the water. So, our water can flip in, it can flip out, it can go back and forth across this cell membrane. Now, we get into the chapter where we deal with osmosis, and we start to focus on how cells are getting water in and getting water out, and what that then does to the cell, whether our cell is going to live, whether it's going to be healthy, or whether it's going to shrivel up and die. We have to keep in mind, we have to refer back to the idea that in the cell membrane, we've got those aquaporins moving water back and forth. So one way to think about a cell membrane, a way that you may be familiar with, is to blow some bubbles. When we blow bubbles, one of the things that happens is the surfactant in our bubble mixture, it starts to form a barrier. It starts to form a boundary. And so here with this little bubble, what we can see is we can see that almost perfectly spherical shape. We've got it defined inside and we've got it defined outside. Essentially, this is like a cell. It's a similar thing with a water balloon, where the balloon itself becomes that cell membrane. Now, the only difference between a water balloon, this bubble, and an actual cell is that these can't really control their interaction with the environment. They can't control what gets in and what gets out. The other thing that they can't do is they can't do any bulk movement in and out. So sometimes cells just want to move individual molecules, and that's where we see our pump protein, our simple diffusion with oxygen and carbon dioxide, or our aquaporin using facilitated diffusion to move water in and out faster than expected, faster than simple diffusion alone. If our cell wants to move hundreds of molecules in at one time, it does something called endocytosis, where the plasma membrane, the cell membrane, starts to fold out and encompass all of those molecules and then bring them inside of the cell body. If a cell wants to release hundreds of thousands of molecules all at one time, it makes all of those molecules on the inside, and then it starts to push that vesicle close to the cell membrane, and then when it reaches, it merges and it just pushes all of those molecules out into the environment. So, if you have ever done any modern dance, you might have noticed that this is a move that we do in modern dance. It's endocytosis, and we have to move it over to biology, and then in dance, when we do that explosive movement, that that is our exocytosis. I know not all of you out there are biology majors, so I wanted to give you something else, another analogy to remember. Endocytosis, bringing in, and exocytosis, pushing out for our forms of bulk movement. We next move into all of the different parts of the cell. Now, one thing that I want to give you as a big piece of advice, please be careful when you are defining the different parts of the cell. I give you a cell study tool to use as you are going through the textbook chapter, as you are going through the lecture slides. And one of the things you're going to notice in the lecture slides is that I get kind of casual in how I define the function of the cell parts. So for example, the vacuole. A vacuole is a plasma membrane sac for storing stuff. I guess you could call it a Rubbermaid container. I'd be okay with that. That's not an inaccurate analogy. Be careful of analogies that don't help you. The lysosome is often called the garbage truck. I hate that one. When we think of garbage, the garbage truck comes, it grabs our garbage, and it takes that stuff away. Now, granted, I know that it goes to the landfill, but in my mind, it just went away. When we look at something like a lysosome, and we look at biological systems, in biological systems, there is no away. There's no final destination. Every output is an input for another system. And so a better analogy for a lysosome would be something like a recycling truck. Because when a lysosome grabs a hold of another organelle, or is trying to break down some sort of engulfed endocytosed molecule, it's going to be breaking it into individual parts, and then those individual parts get reused inside of the cell. So there I'm okay saying that the lysosome is the recycling truck of the cell. Now, as you will see in the previous video and in the lecture slides, I hate it when people call the mitochondrion the powerhouse of the cell. So please revisit the past lecture, the past video, and take a look at those lecture slides for why the mitochondrion needs a better definition. So make sure to get the parts of the cell memorized and their function. The reason why is because later we are going to be taking for granted that you know what the thing is, you know what it does, so we can put it into this larger process of what's happening inside of our cell. The last thing that I wanted to end with today, just sort of a brief explanation, is endosymbiosis theory. Both the chloroplast and the mitochondrion organelle were very strange when researchers started discovering them and figuring out the different parts of the chloroplast and the mitochondrion. They both have multiple membranes. They both have DNA, and that DNA is separate from the DNA that's in the nucleus. That DNA is circular. It's in the shape of a plasmid. And once scientists got a hold of the technology where they could sequence the genes in that circular plasmid in the mitochondrion and the chloroplast organelle, what they noticed was that those genes are more similar to prokaryotic bacterial cells. So this blew their mind because it looks like there's a prokaryotic bacterial cell inside of that larger eukaryotic cell. So this is where we have endosymbiosis theory. Endo being inside, symbiosis being a relationship where at least one of the organisms or one of the entities is benefiting. And so when we look at the mitochondrion and the chloroplast organelle, what endosymbiosis theory states is that we had a eukaryotic cell, it got really close to a bacterial cell, and it started to do that phagocytosing. It pushed out its plasma membrane, that specialized type of endocytosis, and it reached around that bacterial cell and it went rom, 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 and it brought that bacterial cell in. And the weird thing is that instead of killing it, it formed a relationship where both of them. So if we want to model for ourselves, kind of see in real life what endosymbiosis theory might look like, let's pretend this is my eukaryotic cell, and it's huge. Let's pretend this little cat toy is my prokaryotic cell. This is my little bacterium. And to be honest with you, I kind of got the scale right here. Our eukaryotic cell is huge compared to our prokaryotic cell. So our cells are just these sacs of organic molecules inside interacting with one another. And so our plastic bag here with our cell membrane gets really close to our prokaryotic cell. As it gets closer to that prokaryotic cell, it reaches out, engulfs it, brings it inside, and instead of killing it, it's going to form a relationship. So scientists think that this is how it happened for both the mitochondrion and the chloroplast organelle. And they've been able to model this with other bacteria, which is why I've included that picture in your lecture slides. So you can see that we're trying to mimic this to prove to ourselves whether or not this is the way those two chloroplast and mitochondrion organelles came to be. If you have any questions about the cell parts, how cells control their interaction through the environment, and the importance of the cell membrane and the plasma membrane, please